this video we will discuss about how to map and quantify mammalian transcriptomes by RNA-Seq uh, technology. So this is the technology. In simple terms, we will understand how we can quantify different level of RNAs that are present within the samples, especially in the mammalian samples. So this is the roadmap that they have followed, or this is the plan that they have followed. That is, first they stabilize the RNA with the help of this addition of uh, poly-A tails, or we can say the poly-A selections is carried out. After that, this RNA has been fragmented into the 200 nucleotides. Thereafter, it is converted into this cDNA or its complementary DNA sequence via reverse transcription, and thereafter it is converted into the 25 base pair read, and the barcodes are added, such as Nextra or uh, TrueSeq, which are the common bar tags for this Illumina sequencing. After that, these 25 base pairs are being sequenced, and then these are being normalized using different strategies. That is 2 RPKM, 1 RPKM, or this is also 1 RPKM. So this data is then analyzed using different normalized values, such as 2 RPKM, 1 RPKM, and 1 RPKM. And these are the standards normalized unit used to measure transcriptome levels via RNA-seq data. The panel B represents example. That is, this is the example of 1 kilobase genome sequence that has been analyzed by using this strategy. So we can see that this part of the gene is been this part of the gene is been present in, or is been analyzed to a is been analyzed to more time compared to the other parts, and similarly the end part is also analyzed, also found to be more compared to the other part of this 1 kilobase sequence. So this indicates that in this mouse model, the genes that belongs to this sequencing range are more and compared to the genes that belongs to the this middle range of this RNA-seq. And thereafter they also check the efficacy of this model. They check whether this my5c is which is the signature for the muscle proteins of the mouse, is present in is shown in only in the muscles or in the other samples also. And in these results they can find out that the my5c or the occurrence of my5c is more prominent in the muscle cells of the mouse compared to the other parts of the mouse. So this further substantiate the accuracy of this new transcriptome by RNA-seq. Thereafter, in order to validate their reproducibility, they also present some of the data in the panel, in the figures on the right hand side. So in the panel A of this figure, they can show that the technical replicates of the brain RNA-seq data, and we can see that the R-square value is very more in aligned, and it is close to 1. This shows that the technical replicates of the RNA-seq reproducibility of the RNA-seq data is quite high. Thereafter, they also did analyze this data, and it is shown that 93% of the data are exon, and this is also in line with the literature, as most of the data in the mouse or human genome are found belongs to exons, and some of the data are intron, and 3% of the data are intergenic. And then they check the Then they compare this their results, some of their results with the results that are available in the literature or in the library, and they can find out the dynamic range and sensitivity of the data are also in line with the data that are present in the literatures, uh, and we can see that R square value for these data are around 0.99, and they have used this 100 nanogram mRNA to generate this data and compare it with the literature. And when they quantify this data, when they quantify this data, they can see that around this quantification is a function of read number, or that is the whether they are able to produce a reproducible results in terms of the number of reads. And they can see that they have used they have presented the data using different reference values, such as this this value are line for this 27 repeats, and whereas these data are for this 187 repeats, and this orange data is for this 6369 repeats. So we can see that most of their data lies in this range, whereas some of the data are lies in the extreme range, that is around 187 data are lies on this round and lies in this range. So this indicates that compared to this 6369, this number of data that lies that shows non-reproducibility is very low. So this further indicates that their robustness for the quantification, their robustness towards this read count is very good, and can be used to measure, and this strategy can be used to measure transcriptomes in different models or for different experiments. So I hope you have understood something about how these new strategies work and what are the results they have shown to uh, validate their new strategy or new methodologies. If you need to understand more about this transcriptome by RNA-Seq, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.